Hello, I'm Jessica Madigan, Director of Business Development at TriLink Biotechnologies. Today I'm going to be presenting on GMP manufacturing of messenger RNA therapeutics and vaccines and a case study of a self-amplifying SARS-CoV-2 vaccine. I will be covering an introduction to TriLink and manufacturing considerations including raw materials, timelines, scalability, and release assays. TriLink is a contract development and manufacturing organization that specializes in modified nucleic acids. Our product offerings include plasmid DNA, messenger RNA, CleanCap capping technology, oligonucleotides, nucleotide triphosphates, and small molecules. All products are offered as GMP grade or research grade. TriLink manufacturing is guided by expertise in synthetic organic chemistry and pioneering knowledge in mRNA. This understanding enables us to continually respond to customer needs and provide innovative solutions. Our custom-built TriLink headquarters include cGMP manufacturing with cutting-edge capabilities to meet your scalability and capacity needs. We are dedicated to continuous growth and expanding our cGMP facilities in San Diego, California. We have a specialized, stringent quality system and a dedicated team of quality experts to ensure the highest level of consistency, purity, and transparency. We are dedicated to innovation and continuous improvement, making the transition between grades seamless and efficient. GMP manufacturing considerations. When planning your journey to cGMP manufacturing, first it's important to understand that cGMP refers to Current Good Manufacturing Practice Regulations enforced by the FDA under ICH Q7. These regulations provide guidance about good manufacturing practices for active pharmaceutical ingredients under a system for managing quality and operations. They help to ensure that APIs meet the requirement for identity, strength, quality, and purity. You need to consider the grade of raw materials used and the grade of final mRNA product used for your stages of clinical trials. FDA guidance states that GMP grade raw materials should be used in the production of APIs if available. If research grade materials are used, which could include plasmids, NTPs, enzymes, or other critical raws, a risk mitigation approach should be utilized. Regardless of the grade of the material, all raw materials should be tested for identity and sufficiently characterized for ensuring the safety of the mRNA product. The best strategy for production of a therapeutic is to keep it simple. Minimizing the number of raw materials used in the process and the number of manufacturing steps will increase the likelihood of batch-to-batch consistency and maximize your budget. Another important consideration for mRNA clinical trials is qualification of release assays. Methods should be phase appropriate and qualified for use. And finally, manufacturing should be done in an environment that minimizes the likelihood of viable contamination, and all processes should be well documented. The traditional stages of therapeutic development are research-grade material is used for toxicology and preclinical work, and then GMP-grade materials are used for phase 1. One option to condense your timeline is to manufacture a single batch of GMP-grade material for preclinical toxicology use and for phase 1. This can be a high-risk approach from a budget perspective, as the cost of GMP-grade production is expensive, and without relevant animal data, you will not know if your material is effective. After phase 1, manufacturing scale-up is completed to meet commercial or later phase scale needs. The scale-up is first done in a research setting and then validated under GMP conditions. Decisions on the quality of raw materials used in the production of the mRNA should be made early in the development process in order to ensure consistency from toxicology studies to clinical trials. We encourage clients to interface with regulatory agencies early in pre-IND meetings in order to align on the strategy. At TriLink, we offer customizable solutions for your therapeutic development path. Research-grade materials used for screening and selection of your compound of interest. GMP-linked materials are used for toxicology studies, and GMP-grade materials for phase 1 clinical trials. A question that I am often asked is, why do GMP grade products cost so much more than non-GMP grade products? There are several things that add to the cost of most of these GMP manufactured products. Among those are the requirements for QA review, process documentation, stability studies, recurring personnel training, and operation of clean room facilities. On this slide, I'm providing a summary of responsibilities for the client and the CMO. In general, the client is responsible for making decisions as it pertains to regulatory submissions, and the CMO is responsible for providing guidance on the most efficient process to produce the required amounts of material at the mutually agreed upon specifications. The CMO is responsible for manufacturing the product that meets all regulatory guidelines and the required documentation, including certificates of analysis, certificates of origin, certificates of release, and all executed batch records. Moving on to planning and developing a successful messenger RNA program. Selection of a compound of interest. Important considerations for an mRNA therapeutic are sequence selection, tailing strategies, capping strategies, base modifications, and purification strategy. For more details, I encourage you to watch Dr. Anton McCaffrey's presentation on messenger RNA design and manufacturing considerations, a new therapeutic drug class, available on the TIDES on-demand presentations. Process development phase. After selecting your compound of interest, the first phase of GMP program is a process development. The average timeline for this phase is 1 to 6 months. In this phase, the method for GMP manufacturing will be defined, the scale for manufacturing will be defined, and the specification will be drafted. Preceding a successful process development phase is a preclinical phase. Average timelines for preclinical is 1 to 3 months. During this phase, raw material selection will be finalized, master batch records will be drafted, assay qualification activities will be completed, toxicology material will be produced, and stability studies will be started. The final phase of a successful development program is clinical production. mRNA is manufactured under GMP conditions and released for use in humans in early phase clinical trials. Average timelines for production and release of clinical grade products is 1.5 to 3 months. Traditional approach to messenger RNA manufacturing will include the following steps: transcription of an RNA from a DNA template, DNase treatment, and purification to remove process-related impurities from the desired mRNA transcript. The process should take as little time as possible to maximize purity, full-length integrity, and your budget. This manufacturing process can take as little as 1 day or up to 3 weeks, depending on the complexity of the process used and the number of enzymatic treatments. Scalability is also a very important consideration when planning for an eventual commercial production. Innovations in tools, techniques, processes, equipment, upstream and downstream strategies needed to manufacture and scale production of biologics has generally kept pace with messenger RNA manufacturing requirements. Traditional vaccines would require process-dedicated production processes, facilities, and operators. One advantage of messenger RNA therapeutics is efficiency from shared platform manufacturing processes and infrastructure across all therapeutic indications, including gene editing, protein replacement, and vaccines. Bioreactors are often used. They are single-use closed systems. Temperature and pH can be monitored. Scaled down and scaled up models are readily available for preclinical to commercial batch sizes. This will enable a smooth transition to eventual commercial production. mRNA analytics. Testing should be done throughout the process to ensure the quality of the end product. Per regulatory guidance, all raw materials entering a GMP manufacturing process must be tested for identity. Additional testing is carried out based on risk. For example, if a research-grade material is being selected for use, it may be important to test for purity, functionality, or viables before using in manufacturing. In-process testing for yield, purity, and safety should also be included. Furthermore, monitoring or controlling for critical process parameters such as temperature, time duration, and pH are important for quality assurance. Finally, the product should be tested and per agreed upon specifications. Developing specifications for an API require comprehensive and critical analysis of the requirements that are used in developing and qualifying sound analytical methods. The approach to setting specifications should be process-driven in order to meet both the FDA and ICH standards. In early phase of clinical development, specifications should be based on the results of toxicology studies that provide purity limits. As development advances with clinical studies, the specifications should evolve to encompass tighter acceptance limits based on the manufacturing capabilities and stability data. Once submitted to the FDA, the specifications may need to be revised. That would require a new round of review and approval of the revised specifications by the quality, analytical development, and regulatory functions at CMO and at the client level. When selecting assays, the absolute minimum panel of release testing should include ID testing, strength testing, impurity testing, and safety testing. Release assay panels and specifications should be submitted to the FDA for review in a pre-IND meeting to maximize the likelihood of regulatory acceptance. The most common types of messenger RNA release assays are shown here. Identity test methods include sequencing, in vitro translation, RNase digestion, RT-PCR to detect amplicons of a specific size, or fingerprinting. Process-related impurity testing includes residual protein, residual DNA, and double-stranded RNA content. mRNA purity is often measured by capillary electrophoresis, fragment analyzer, bioanalyzer, size exclusion chromatography, or ion pairing reverse phase chromatography. Concentration is measured by UV spectrophotometry. Messenger RNA capping efficiency and tail length are also often measured as they directly correlate to the potency of the mRNA product. USP assays are used for physiochemical properties and for safety. Assay qualification. The objective of a validation of an analytical procedure is to demonstrate that it is suitable for its intended use. Documentation of analytical methods forms a key part of the specifications. Pharmaceutical manufacturing continues to evolve with advanced technology, tools, and processes for sound analytical methods and testing. Effective use of the most current pharmaceutical principles and knowledge, which is documented in specifications that are carried throughout the life cycle of an API, can lead to faster commercialization as well as safe and effective products. On this slide, you will find an example of a general approach to a method qualification, which is required for phase 1 clinical trials. Methods can be tested for accuracy, precision, specificity, limit of detection, limit of quantification, linearity, and range. When putting together your program timelines, you will need to include 6 to 12 weeks for method qualification. And in order to qualify a method, you will need representative material. The strategy that is most often employed is to use material that is produced for preclinical toxicology studies to qualify the analytical methods for phase 1 GMP production. Setting specifications. I caution clients to set specifications that are achievable and also based on efficacy results. Do not set your specifications too stringent, as you will want to continue to tighten and refine your specifications as you progress through your clinical phases. Additionally, I cannot stress enough the importance of establishing a relevant clinical model as early as possible. In vitro models do not always directly correlate to an in vivo response. Different routes of administration will elicit different innate immune responses, which directly correlate to the potency of your therapeutic. Specifications will also depend on the existing technological state of the art. For example, it is unrealistic to expect that a messenger RNA that is greater than 5,000 bases to meet a greater than 95% purity specification by a bioanalyzer. So when setting a specification, it's important to consider the length and the state of the art of the technology. When setting a batch size for production, it is also critical to consider the amount of material that's going to be required for final release and retention samples, as oftentimes the amount needed for quality control will exceed the amount needed for an actual clinical study. In the next few slides, I will provide examples of specifications for the release of a messenger RNA therapeutic for phase 1. Please note that this panel is being presented as general guidance. Sequencing of an RNA product by RT-PCR followed by PCR followed by Sanger sequencing is often used for identity. The specification is set that the results will conform to the theoretical sequence. Visual appearance of a messenger RNA is expected to be clear, colorless, and free from visible particulates. The length of the product should be confirmed, either by fragment analyzer, bioanalyzer, or other type of CE instrument, and it should match the theoretical length plus or minus 10%. Concentration is measured by UV absorbance at 260 nanometers. Concentration should be plus or minus 10% of the target value. pH of an mRNA is generally 5 to 7, but this will be dependent on the buffer composition. Osmolality can also be important for downstream manufacturing, especially for ex vivo applications. Osmolality of an mRNA in water is generally low, but also please note that the osmolality will be dependent on the buffer composition. Integrity of an RNA product can be measured by smear analysis on a fragment analyzer. The percent purity achievable will be highly dependent on the length of the construct. The shorter the sequence, the higher the purity can be achieved. The specification for purity should be correlated to a functional potency and should be as broad as possible for phase 1 clinical trials. Bioburden and endotoxin specifications should be set based on intended maximum dosage of the therapeutic. Less than 1 CFU per mil and less than 3 EUs per mil are generally achievable for a messenger RNA manufacturing process. Residual proteins and residual DNA should be measured. Less than 2% weight-to-weight for residual proteins and less than 1,000 picograms of DNA per mil of RNA are example specifications drawn from the World Health Organization's guidance for biological products. Again, these specifications should be set based on your final product dose strategy. Residual solvents should be measured and adhered to safety limits. The solvents tested should be driven by the manufacturing process. Capping efficiency should be measured and specifications set based on a correlation to the potency. Greater than 90% capped is recommended. Double-stranded RNA is a potent innate immune stimulator. Due to its adjuvant properties, it may be a desired impurity depending on your therapeutic indication. It should be measured and monitored for batch-to-batch consistency. For all regulatory filings, at least 1 month of stability data is required for submission and approval. The purpose of stability testing is to provide evidence on how the quality of a drug substance or drug product varies with time under the influence of a variety of environmental factors, such as temperature, humidity, and light. Additionally, it is used to establish a retest period or shelf life of the product and the recommended storage conditions. For mRNA drug substances with a proposed retest date of at least 12 months, the frequency of testing at long-term storage conditions should normally be every 3 months over the first year, every 6 months over the second year, and annually thereafter through the proposed retest period. At accelerated storage conditions, a minimum of 3 time points from a 6-month study is recommended. Also, I wanted to note the thermostability of messenger RNA. mRNA can be surprisingly thermostable. Extensive testing of mRNA storage has shown good stability in liquid form when the product is refrigerated or frozen. Especially when formulated in a messenger RNA LNP complex, it has been shown that mRNA is stable at 4 degrees C and 25 degrees C for at least 7 days. Other studies on lyophilized messenger RNA products have demonstrated stability for 24 months at ambient temperature. I hope I have provided you with a greater understanding of the important considerations for a GMP manufacturing campaign. And now I'd like to present to you a case study timeline for a rapid vaccine development of a SARS-CoV-2 self-amplifying messenger RNA vaccine. First, it's important to note, to accelerate vaccine manufacturing timelines, it's important to streamline the manufacturing process development. By utilizing a well-developed platform manufacturing process and reducing the number of manufacturing steps, timelines can be significantly compressed. For the fastest possible manufacturing approach, we recommend a templated poly-A tail in the plasmid, a co-transcriptional capping method, such as TriLink's clean cap technology, and a one-step purification process. Researchers around the world are developing more than 165 vaccines against the coronavirus, and 32 vaccines are in human trials. Vaccines typically require years of research and testing before reaching the clinic, but scientists are racing to produce a safe and effective vaccine by next year. Imperial College London has developed a self-amplifying vaccine for the SARS-CoV-2. Work began in January with deciphering the SARS-CoV-2 genome. It was at that time that TriLink was engaged as a potential CMO partner. The scope of work was issued and agreed upon. A 5-day manufacturing process was designed from plasmid linearization to final processing. The saRNA was produced and shipped for LNP formulation under quarantine while quality control analysis were completed. Toxicology studies were initiated while GMP manufacturing proceeded at risk, and GMP manufactured products were shipped under quarantine for LNP formulation. This program was able to condense an average 6-month timeline to just 63 days. The Imperial College London vaccine, which boosts production of a viral protein to stimulate the immune system, began phase 1 and 2 clinical trials on June 15th. Researchers are expected to know if the vaccine is effective by the end of this year. At TriLink, we strive to exceed your expectations by maintaining the highest level of quality and service. We believe it is our responsibility to work with you to push past technical barriers and create solutions. Our product management, manufacturing, and quality assurance teams work together to understand your challenges and deliver the best possible therapeutic products. Thank you for your attention today, and I welcome any questions.